Hi everyone, in this video I'll be explaining how the polymerase chain reaction works. This method, also known as PCR, is used to make many copies of a specific sequence of DNA. It is widely applicable and has lots of practical uses ranging from forensics, genetic testing, and diagnosis. PCR is very sensitive and can detect the specific sequence of DNA or gene of interest even if it's present in only tiny quantities. So here we have our DNA that we want to be amplified. This is mixed with deoxyribonucleotides, or DNTPs, a thermally stable polymerase from a thermophilic bacteria called Taq polymerase, and our DNA primers. The DNA primers hybridize to the ends of the gene to be amplified and provide a starting point for the Taq polymerase to start replication. In the next step, the mixture is then heated to 95 degrees Celsius to break the hydrogen bonds in DNA, forming single-stranded DNA. The mixture is then cooled to 50 to 65 degrees to allow the primers to bind to the complementary regions on the DNA. These primers are chosen specifically so that they flank the gene of interest. Once the primers have binded the DNA, the temperature is raised to 65 to 75 degrees, allowing Taq polymerase to bind the primer and synthesize the complementary DNA strand. Next, the cycle repeats and the temperature is then raised again so that the DNA strands separate. Once the DNA strands have separated, the temperature is lowered so that the DNA primers can bind again, and Taq polymerase can synthesize another complementary strand. This process is repeated until enough DNA has been produced for the particular experiment. After 30 cycles, PCR can amplify one molecule of DNA into 10 billion copies. This kind of amplification is achieved by running the reaction in a thermocycler, which automatically raises and lowers the temperatures at appropriate time intervals.